what is nutrigenetics? So identifying a phenotypic, an observable trait response to a dietary intake that is determined by the genotype, genetic makeup of an individual. And we could think of a therapeutic goal, this overall, what are we building toward here? And it would be to create targeted dietary recommendations for disease treatment and prevention based on an individual's unique genetics. When you hear nutrigenetics, there's also these related terms, nutrigenomics, which, as the name implies, is similar, except instead of looking at the genetic level, we're looking at the genomic level and the epigenomic level, as well as personalized nutrition, precision nutrition, and tailored nutrition. And I'll go through those terms in a moment. There is a growing interest in nutrigenetics. So over the past 20 years, we could actually say that this is a fairly new field. And over the past 20 years, we've seen what's essentially a linear increase in the number of publications, which is great. There's a growing interest. It's a burgeoning new field. And when we're looking at the applications of nutrigenetics, it's useful to think of them in terms of resolution. So we could start at the lowest resolution. So this would be like recommendations for large effect conditions. So I took some terms from Ordovas 2018, but these are all very new terms. So there's no real consensus on what these terms exactly mean at this point. But I like this definition, so I used it. So tailored nutrition, these broad scale things like PKU, lactose intolerance. We have a straightforward implementation, right? Don't eat dairy, don't eat phenylalanine, right? And it's commonly used and employed effectively today. So this is already happening. And then we can think of medium resolution, so recommendations for these small to medium effect nutritional susceptibilities. So we could call this personalized nutrition, things like, take a general demographic group. So for example, European women age 45, right? And they have a mutation at minor allele RS1234, right? And maybe they're at increased risk of iron deficiency because this is a mutation in one of the iron enzymes, right? So this would be like a medium resolution. And then the high resolution, this is like the dream, is a quantitative or even algorithmic understanding of the complex relationships in the human organism. We could call this precision nutrition. So an individual, Mary Beth Johnson with haplotypes A, B, C, D should eat 42% carbs, 25% fat, supplement this, don't supplement that. Maybe if you're exercising, you should have exactly this much more, right? So that's kind of like the pipe dream. But as of right now, in my estimation, I think we're right about here, right? So we're just entering this medium resolution domain of nutrigenetics. And there are some challenges in implementation. And especially there's some challenges to get to that high resolution understanding. For one thing, we're dealing with small effect sizes and variance, right? So I'll talk about this in the context of GWAS in a minute, but we're looking at variants that have small effects. So there's not one gene that dictates what you should eat, right? There's many small effects throughout the genome. And these biobank scale data sets have actually only recently emerged with adequate power to get to this medium resolution, right? I'm thinking of UK Biobank. It's like we're just getting enough power to get to that level. There's also been few randomized control trials to date in terms of nutrigenetics research, although I hope that that changes in the near future. And then there's these hidden factors which can confound nutrigenetics, making it even more complicated. Microbiome, resting metabolic rate, stress, breathing, right? Environment, maybe you live near some pollution. So there's a million things, right? And when I was studying nutrition in my master's, there's a phrase which is studying nutrition or optimizing nutrition is like trying to balance an 8-sided teeter-totter. It's like you have more of one thing and then you're having less of another. And it's this very delicate balancing act. So in terms of the nutrigenetics development pipeline, I kind of think of this similar to the drug development pipeline. And we could think of, from the beginning to actually being used by people in the population, right? So disease characterization, this would be like ICD-10, right? You have to know what the disease is before you can study it. And then variant discovery, which is where we live. And then we pass our results to maybe some cellular studies or some animal studies. And then eventually, once you get validation in that way, you can go to randomized control trials. And finally, maybe the FDA is going to say, hey, you should do X, Y, Z, right? But we live right here in variant discovery. And if we want to zoom in to variant discovery a little bit more, we could think of the steps, which are collecting the genotype and phenotype data. And this is the one part that we didn't do. This was done by UK Biobank, so it's really cool. So we received that, and then we did the rest of this. So this is what I've been doing, is designing analyses, quality controlling the data, conducting statistical tests, replicating and meta-analyzing the results, and then doing some follow-up, putting our results into context. And so I'll introduce this tool, UK Biobank, now because it's what we used as the primary data set for these studies. So UK Biobank is a publicly available health research database. It's fairly new, only the last 3 years. And it collected the health status and genotype of 500,000 volunteer participants. They were aged between 40 and 69 years old in the years that this was active, 2006 to 2010. 88% white British, which is unfortunate limitation, right? So these results are really mostly only generalizable to Caucasian population. This is a general problem in quantitative genetics, which is being fixed currently. And then one really cool thing for us as nutrigenetics researchers is that UK Biobank has 2 dietary surveys. They have a food frequency questionnaire that they gave at the initial assessment, and a 24-hour recall survey, which was administered in voluntary waves to these participants. So that's our data set. Let's start to talk about GWAS and the theory behind that. So we have what's known as the common disease, common variant hypothesis. And I say this every time that people in my lab have seen me explain this at least 20 times, but I'll do it one more time today. So the idea is that the genetic component of common disorders is due to a large number of disease-causing alleles that occur often in the population. So in contrast to a Mendelian mutation in the upper left quadrant, things like cystic fibrosis, cleft chin is one of these, where it's like one gene is directly impacting a phenotype. We're more interested in this lower right-hand quadrant, where we have many small effect variants throughout the genome that are sort of giving this cumulative phenotype, right? So things like some certain types of cancer, diabetes, obesity. This is why you never hear of the obesity gene, right? There's no one spot in the DNA that makes you obese or susceptible to that. But what we have is, we can develop a polygenic risk score with, let's say, 80 variants that are making some contributions to obesity throughout the genome. And so the tools we used were GWAS and GWIS. These are related statistical analysis techniques to identify significant nutrigenetic variants. These are single nucleotide polymorphisms when they're a single base pair of DNA. These are called SNPs, but I use the term variant because we're also talking about indels, which can be multiple bases, right? And genome-wide association studies are used to find associations of allele frequencies with disease traits. And we're interested in the effect or the odds ratio or the p-value of a variant, whereas in an genome-wide interaction study, we're really interested in these gene-environment interactions and the effect or the odds ratio or the p-value at the interaction. So what does all that mean? Well, it's really, the statistics are not incredibly complicated. We're really just doing about 1 million regression models for each spot at the DNA. So for every base pair that's been genotyped, we're running a regression model. And so we could think of it as the trait of interest, Y would be like the dependent variable in the model. And then we could code the minor alleles. So remember, you have 2 copies of DNA, right? So you can either have 0, 1, or 2 copies of a minor allele. Throw some covariates in your model, and that is your GWAS model in a nutshell. For GWIS, we are adding this sort of higher order statistical interaction term where we can, take what would have been an ordinary covariate and call it the environmental exposure. So, maybe this is the dietary intake of interest, or whatever, and then look at the interaction between that and the genotype term in the regression. So that is how they work. And so like I said, we're interested in this beta GG in GWAS, so the effect of the genotype term, and then beta gene environment G by E, so the effect of the interaction, right? So in terms of gene environment interactions, there's actually many different ways that these could happen biologically. So for example, your genetic variant influences the enzymatic metabolic efficiency of a food or a toxin, or a variant modifies the risk benefit of a behavioral exposure like exercise. You could think this person only exercises for 2 hours a week and they're, ripped, and I exercise, double that, but I, whatever, right? And then you could think of another way, which is an environmental exposure modifies gene expression epigenetically. So this is actually what PUFAs do, right? So PUFAs can epigenetically impact gene expression. So there's a lot of different angles that these interactions can happen. I may be biased, but I think that diet is the most important environmental exposure to study. And I think that because of 2 reasons. For one, it's a fully modifiable factor, right? So you can completely, within limits, but it's really modifiable. You could change it a lot. And it also, the food you eat directly interfaces on a cellular level with the organism, right? So it's this really direct impact that we have essentially full control over. So I think it's a really nice field to study gene environment interactions. Our tool for visualizing the results of a GWAS and a GWIS is the Manhattan plot. This is the main visualization tool for finding these results. And so on the x-axis, we are looking at every variant by chromosome that has been genotyped. And on the y-axis, we have the negative log of the p-values. So in short, the higher the peak goes, the higher the points go, the more significant these points are going to be. This red line indicates our typical genome-wide significance threshold of 5 × 10⁻⁸. And this is derived from the classic alpha level of 0.05 divided by 1 million LD blocks throughout the genome for multiple testing. We can zoom into one of these peaks and get a regional Manhattan plot. So we can kind of determine exactly which variants are involved in one of these peaks. And so in this case, we have a lot of genetic variants that are in linkage disequilibrium with each other. And so this is kind of the struggle of doing GWAS, is that you're really running many regression equations, but the variables in the regressions are highly correlated with each other, right? So this is what we're doing. So we think it could be in terms of causal variant, we think it's the highest p-value, but maybe it's not, right? So we have to kind of consider this entire block, this LD block. And when it's a significant block like this, we can call it a genomic risk locus. Typically presented with Manhattan plots are these QQ plots, quantile plots. And so these allow us to visualize the distribution of the p-values as compared to a chi-squared distribution. So essentially what we can see in a QQ plot is whether we have systematic inflation in our p-values or whether it's following our expected distribution. So these are also typically shown with the Manhattan plot. So the diagonal is our expectation under the null hypothesis. And then above the line would be either true causal effects or inflation, right? And this is something we have to deal with as researchers.